highly muscular wall controls the blood. Their walls are divided, leg in leg, then triangle, thick, uniform medium, circular, smooth, and round. Parenchyma is lined by simple squamous epithelium, a thin subendothelial layer, and a prominent wavy internal elastic lamina. Function provides a smooth surface, prevents clotting, and separates the intima from the media. Tunica media, thin layer containing 3 to 40 layers of spiraling smooth muscle cells. That is thick layer. Containing of 3 to 40 plus layers of spiraling smooth muscle cells interspersed with collagen and elastic fibers, bounded outwardly by an external elastic lamina. The regulate lumen caliber, direct blood distribution, and maintain blood pressure. Tunica adventitia, dense irregular connective tissue, including collagen fibers, fibroblasts, and macrophages, contains tiny blood vessels, vasa vasarum, and nerves, nervi vasarum. It is the artery to surrounding tissues and provides nourishment to outer wall layers. So, some more slides of muscular artery histology. Next is elastic artery histology. Elastic arteries, for example, the aorta, pulmonary trunk, are large conducting vessels that dampen blood pressure fluctuations. Their walls are organized into three distinct layers: the tunica intima, tunica media, and tunica adventitia. Tunica intima, the innermost layer. Endothelium, simple squamous epithelium with dark staining, flattened nuclei facing the lumen. Subendothelial layer. Connective tissue containing fibroblasts and myointimal cells. Internal elastic membrane. A fenestrated band of elastin. In elastic arteries, it is often difficult to distinguish from the neighboring media. Tunica media, the middle layer. This is the thickest layer. Elastic lamellae. Features concentric fenestrated sheets lamellae of elastin. Adult aortas can have 40 to 70 of these sheets. Smooth muscle and extracellular matrix. Smooth muscle cells and collagen fibers lie between the elastic lamellae. Function provides the Wind-Kessel effect, stretching to absorb systolic pressure and recoiling to maintain diastolic pressure. Tunica adventitia, the outermost layer, a relatively thin layer of dense irregular connective tissue. Vasa vasorum, small blood vessels that supply oxygen and nutrients to the walls of large arteries. Nervi vasorum, autonomic nerve fibers that innervate the vessel wall. These are seen in tunica adventitia. Some more slides of elastic artery histology. Next, vein histology. Under the microscope, veins are thin mold capacitance vessels that characteristically appear collapsed, irregular or floppy in shape, with a large wide lumen. Unlike arteries, they experience low hydrostatic pressure, which is directly reflected in their histological composition. Veins share the same basic structural framework as arteries, consisting of three distinct concentric layers called tunics. Tunica intima, the innermost layer, tunica media, middle layer, and tunica adventitia or externa, outermost layer. Three layers, tunics. Tunica intima contains endothelium composed of a single layer of simple squamous epithelial cells lining the lumen. It functions as a selectively permeable barrier and presents a smooth surface to prevent blood clotting. Subendothelial layer of the tunica intima. A very thin layer of loose connective tissue containing sparse collagen fibers. Internal elastic amina, absent or highly distinct in most veins. Only medium-sized veins occasionally show faint, poorly defined. Tunica media of veins composition contains a relatively thin layer of circumferentially arranged vascular smooth muscle cells interspersed with collagen and elastic fibers. Thickness much thinner than the corresponding layer in arteries. It typically comprises only 2 to 8 layers of smooth muscle cells. The external elastic lamina absent or indistinguishable. The tunica dentitia veins, tunica externa, significance this is the thickest and most prominent layer in the walls of veins. Composition predominantly composed of dense irregular connective tissue, rich in type 1 collagen fibers and networks of elastic fibers. The tunica dentitia veins. Internal elastic lamina, absent or highly indistinct in most veins. Only medium-sized veins occasionally show a faint, poorly defined IEL valves. IEL valves formed by semilunar projections of the tunica intima. They consist of a core of connective tissue covered by endothelium and are highly prevalent in the veins of the lower extremities to prevent the retrograde flow of blood. Tunica media composition contains a relatively thin layer of circumferentially arranged vascular smooth muscle cells interspersed with collagen and elastic fibers. Thickness tunica media much thinner than the corresponding layers in arteries, typically comprise of only 2 to 8 layers of smooth muscle cells. External elastic lamina absent or indistinguishable. Specialized features. Vasa vessel. Vessels of the vessel. Because venous blood is low in oxygen, large veins require an extensive network of small blood vessels in the adventitia to supply oxygen and nutrients to the outer layers of the vein wall. Nervous vasorum, autonomic nerve fibers regulating minimal vasomotor tone, the vessel. Longitudinal smooth tissue. In large veins like the vena cava, the tunica adventitia contains prominent bundles of longitudinally arranged smooth muscle to help propel blood against gravity towards the heart. Some more slides of vein histology. Capillaries histology. Capillaries are the body's smallest blood vessels, 5 to 10 microns in diameter, acting as the crucial exchange sites between blood and tissues. They consist exclusively of a single layer of simple squamous endothelial cells surrounded by a basement membrane and scattered pericytes. Macroscopic structure endothelium, a single layer of flattened epithelial cells forming the tube. It makes up the tunica intima, allowing for the rapid, efficient diffusion of gases and nutrients. Basal membrane, thin basal lamina that wraps the endothelial cells, providing structural support. Pericytes, contractile stem-like perivascular cells that partially wrap around the capillary. They help regulate blood flow, maintain the blood-brain barrier, and aid in tissue repair. Three structural types: continuous capillaries, and sinusoidal capillaries. Continuous capillaries, the most common type, featuring tightly joined endothelial cells with no pores, found in muscle, skin, lungs, and the blood-brain barrier. Fenestrated capillaries, second one. Endothelial cells contain tiny pores, fenestrae, that make them highly permeable to fluids and small solutes, located in endocrine glands, kidneys, and intestinal villi. Third one is the sinusoidal capillaries, discontinuous, wide leaky gaps between endothelial cells and an incomplete basement membrane found in liver, leg, etc. So, some more slides of capillary. The capillary. You can see the capillary is lined by a single layer of endothelial cells inside RBCs. You can see. So, you can see the capillary. Today, have discussed the histology of blood vessels. Thank you.